Good afternoon, everyone. My name is Pratik, and I'm excited to walk through my research on epigenetic modifications, Alzheimer's disease, particularly how these modifications can be targeted for Alzheimer's disease therapy. So to start, I started by considering the research questions that basically helped me form the basis of my entire study. And one question was, what specific epigenetic modification or epigenetic genetic modifications are associated with Alzheimer's disease, and what potential therapeutic approaches exist that target those specific epigenetic modifications. And if anyone is wondering why this research is so important, well, Alzheimer's disease is a growing public health challenge, not just in the USA, but throughout the world, and currently affects about 6.5 million people aged 65 years or older just in the United States, with that number expected to double by 2050. So, moving on to my literature review, I focused on key epigenetic modifications related to Alzheimer's disease. And one epigenetic modification I focused on was DNA methylation. And an example of that is reduced levels of 5-HMC, which is basically a DNA pyrimidine nitrogen base derived from cytosine that has extensively been linked to Alzheimer's disease and has caused many neuronal deficits and increase in production of amyloid beta, a major hallmark, I would say, for Alzheimer's disease. Then another epigenetic modification I focused on was histone modifications, and changes in histone marks such as H4K, 16AC, as I mentioned, are associated with Alzheimer's disease. And finally, in terms of RNA, I focused on two different RNAs, microRNA and long-coding RNA, with microRNA such as microRNA 132 having major deficits in memory, which correlates with Alzheimer's disease, and another type of RNA, long non-coding RNAs, which have been found to show abnormal levels of RNA expression like MALAT1 and BASE1AS is observed in Alzheimer's disease. And targeting these RNAs could help reduce inflammation and support neuronal health, which is beneficial for as a therapy for Alzheimer's disease. And as for my methodology, I conducted an extensive literature search using various databases to gather relevant articles and studies focusing on epigenetic biomarkers and therapies related to Alzheimer's disease. And once I've selected various articles, I performed a qualitative analysis to identify patterns and key insights regarding my topic. And in terms of my results, first, I found that DNA methylation inhibitors such as ECGC work to improve cognitive functions and have shown to alleviate AD pathology or progression in mouse models. And they come in the form of drugs or compounds. And an example is such as ECGC is found in green tea and show promise as potential therapies for AD. And another inhibitor that I focused on, which was histone deacetylase inhibitors, also known as HDAC inhibitors, have shown major improvements in AD mouse models and cellular models. For example, sodium valproate, which effectively helps reduce amyloid beta depositions and improve cognitive functions in those mouse models and AD cellular models. And finally, for non-coding RNAs, targeting specific RNAs and changing their expression or by overexpressing them or de-expressing them, those RNA molecules, they offer another potential therapeutic approach for addressing abnormal RNA levels in Alzheimer's disease. And in conclusion, my findings showed that Alzheimer's disease is associated with abnormal DNA methylation, histone modifications, and microRNA and non-coding RNA changes. And their subsequent epigenetic therapies have also shown potential as a therapeutic approach for AD, and those therapeutic approaches include DNA methylation inhibitors, HDAC inhibitors, and non-coding RNA alterations. And for my contribution, my research highlights critical epigenetic changes and evaluates potential therapies for Alzheimer's disease, offering a foundation for future studies and therapeutic development. And finally, I would like to thank Dr. Varkey for helping me understand the concept of epigenetics better through my one-on-one classes and helping me form a good structure for my overall essay. And I would also like to thank Professor Virgil for helping me throughout this entire process in terms of structuring my entire essay and helping me fix any grammatical or any structural problems I had throughout my entire writing journey. And thank you.
Great job. We can stop presenting before Dr. Jobin comes in. This is a very nice topic. How do you intend on taking it further?
So in terms of taking it forward, I would like to do more intensive research on why this specific epigenetic therapies has not been moved forward into human clinical trials, and there's kind of a halt in preclinical trials in mouse models and cellular models, which I would like to research and see if there's any limitations in terms of the research itself or any studies.
Okay, great answer, but that's a Dr. Varkey answer. Sorry, my question was your studies. It's pretty important. So you could perhaps look for a geriatric expert around your area, and we will teach you how to approach them. You don't say, hey, can you please mentor me? You say that, hey, I've done this groundbreaking research, which is fairly interesting, and I want to share those results with you. It might help you in your treatment. We can like get together or something like that, and you take your research and knock on their doors.
Okay. Yep, I could definitely
because
do that.
you've got to get out of the four walls and share. Like I said, Mona Lisa is not to be kept in the basement. Got it? Yeah, great job. Go ahead, Dr. Varkey.
Good job, Pratik. Like a very good presentation. So it was very detailed. I have a specific question for you. So we know, okay, that if you affect the DNA methylation and all, like it can eventually result in either like development of Alzheimer's or repetics. You can try to prevent them. But what does DNA methylation or histone methylation? What is that methylation or demethylation? What does it actually do?
So they basically alter so the whole point of epigenetic mechanisms such as DNA methylation, histone modifications, as I mentioned, they directly alter the gene activity through environmental factors and many other factors without really altering the DNA sequence itself. So by altering gene expression and gene activity, it basically is a major role in AD pathology and progression in the human brain.
Yeah, so yeah, that's a right answer, that it changes the gene expression and it changes by unwinding or winding chromatin.
Yeah.
So it makes the DNA sequence available for transcription factors to bind onto. Or the other way, like it tries to, condense it so that the DNA sequence is not accessible for expression. So maybe you can include that in your presentation too, whenever you, because some of the individuals who are not familiar with this particular point, they might not know what it actually does. But very good presentation, Pratik, and then continue to develop it more and look more into what goes on in Alzheimer's. Congratulations and best wishes for your future.
Thank you.
Excellent job, Pratik.